Hello everybody. In last lecture I talked about human vocalization or human speech production, and I mentioned that I will talk about the song production by the birds. That means how the songbirds produce songs, which is very similar to human speech production. So understanding the neural mechanisms of songbirds' brain function would help us to understand the human speech production. Okay, and if you see how the brain produces the speech, you will be able to understand how the sensory inputs are processed, then how the memory system creates templates, and how the motor system sends signal to the muscles and vocal cords to control and produce the speech or song. So all those important components are there: sensory inputs, then integration right in the brain, and also you can understand how the memory system works and motor output is created. So it gives you kind of a big picture in one thing. Anyway, the neural network in the songbird, if you see, there are many discrete brain regions that are interconnected in the brain. They form circuits, and there are distinctive circuits in the brain of the songbird, and each circuit is responsible for particular functions. Okay, so we'll see that what are the important discrete areas and how they are connected and their important functions. A lot of research is being done on the songbirds' brain, and still a lot of things are not clear. However, we already know a lot of important things about the brain of a songbird. Two things are highly specialized in the songbird. One is the brain that I mentioned, that the structures, specific structures in the brain, and those structures together form the song system. And also the respiratory tract is well designed to produce varieties of songs, and each species produce a particular type of song, but the song is highly complex. Okay, there are motifs, then within each motif you will find the syllables, and within each syllable you will find the notes. So notes are the smallest units of song. Okay, during the learning of language or speech, how to produce song by the songbirds, two templates are used. One is the innate template, another is the acquired template I mentioned in last class. And in innate template you will find the structures. Those are developed by the guidance of the genes, that means genetically guided. So those structures or parts function based on the species and their genes. and that part, innate template, is kind of fixed. However, acquired template is developed by the learning. Okay, so innate forms a foundation on which the learning develops the acquired template. Okay, so innate is genetic or by nature. Okay, and this is by training or learning or nurturing. So during the learning of a song, the songbird, the pupil or the learner first would listen the parent's song. However, structures already are developed in the brain. Those must be present to learn the song from the parent. 3 steps, as I mentioned before, that first sensory input via the auditory system, and then processing where the memory system works, and then motor output goes to the vocal cord and other respiratory tract structures. Okay, so tutor produces the song, and that is listened by the pupil, and in the pupil's brain all these functions take place: integration and memorization, then vocal or motor output. Okay, this is the spectrogram you see that is showing the songs of the bird, and there are different stages of song learning. So there are 5 stages. First, there is critical learning period when it must hear the song of its own kind. That means during the critical period, the pupil or learner would listen the parent's song. Okay, so that is that period. And if you record the sound produced by the learner during the critical period, it's like no articulated, no structured, or no meaningful sound. Okay, like human baby, when just in very early stage, the infants produce the sound like that. Then next stage is the silent period, and during the silent period there is a silent period, often several months, during which those songs are simply stored in the memory. So during the silent period, the memory system creates templates of the parents, and then the next stage is sub-song stage or period. During this period, the pupil will or learner will start to try to produce a song based on the memory template, and this time the song is not yet structured, kind of soft rambling version of the song. There are some elements there, but not crystallized or structured yet. It's much the way a human baby babbles. Then the plastic song period. This period is close to perfect song stage. That means you will see the structures in the song clearly in the spectrogram or sonogram, and in this period, although you will see the structures, but these structures can be changed or modified Okay, if you change the environment, then the song can be changed. You can, it's like flexible, so you can modify the song. That's why it is called the plastic. That means during this time you have the flexibility in the song. And then the crystallized stage, which is the most mature stage. So you will see pretty solid structures and articulations. So this is the crystallized stage, and you can see this is the tutor song, parents song, and this is the crystallized stage of the pupil's song. So you can see very similar. If you see these structures, very similar to the parents song. Makes sense? So during this period, once the crystallized period is passed, or arrives, then you won't be able to change that song much. The flexibility or plasticity is no more there. Okay, so those are different stages or periods of the song development in the songbirds. And during all these steps, you will see that how different brain structures participate or perform their functions. This is very interesting that at different stages, different uh, discrete areas and their connectivities work. Okay, and a lot of experiments have been performed during different stages of development. So that would give you a good idea that how different brain structures work at different stages, perform different functions. Okay, if you see in general a songbird's brain and a non-songbird's brain, it is very interesting that you see this is a non-songbird's brain, only few structures, right? And most of those are in the lower brain parts, and mainly vocal center, and from there the output. So you see the outputs are going to the respiratory muscles and syntax. Syrinx. Syrinx is the lungs or respiratory organs, which is highly capable to produce many different notes in a short period of time. Our lung cannot do that, and syrinx is very interestingly designed, so it can keep one side on and another side off, and alternate on off one side to another side, quickly can switch, and that way they can produce many notes in a short period of time, many more than the human, because they can close and open alternatively to syrinx. Anyway, so you see in non-song bird's brain, it is like motor output from the vocal center in the brain, and motor outputs control the respiratory muscles and the syrinx. Okay. However, if you see the brain of a songbird, you will see a number of structures are involved, and a number of networks are involved, because song learning and producing the song involve a lot of work, lot of activities, right? So this part is innate. The birds, non-song birds, they don't need training, they just produce the call, and already it is developed in the brain genetically. So no learning is needed in this process. No memorization is. However, in case of songbird's brain, there are different systems or connectivities. One is responsible for the song production, another is responsible for the sensory processing, another is responsible for memorization and practice, crystallization. All those things must occur right in the brain. So that's why you see many different areas in the songbird's brain. How the studies are performed to understand different structures in the brain. One way is just injecting dye that will travel through the axon right to the next structure, and if you just inject the dye in one structure and then dissect the brain and try to find the pathway of that dye, that would give you the information how that area is connected to other areas, right? So that is one way, and you can actually illuminate the dye stained cells and fibers. There are many different ways we can do that. Another is inactivation. This is very helpful if you permanently or temporarily inactivate by injecting like inhibitor neurotransmitter or some chemicals that will destroy the area. Both are used to study the functions of different areas. When you inactivate that area and see the behavioral change of behavior of the bird song production, you can figure out that which area is responsible for which functions. Those areas are responsible for learning. When you inactivate those areas, the learning will be significantly hampered. Learning process will be delayed or stopped. However, once the learning is completed in the adult bird, if you inactivate the learning system or the structures responsible, those are responsible for learning, you will see that the bird can still produce the song because learning is already completed, right? So after the learning is completed, if you destroy or inactivate the learning system, that would not impact the song production, right? Another interesting finding is that during crystallization, as you have seen in last slide that I showed different periods or stages, during the crystallization occurs, there is a stage that is called the plasticity stage, and during that period, if you inactivate, then what will happen, the crystallized song will not be formed, or it will be delayed. Okay, so different ways are used to study the learning of and producing the song. Another is stimulating one area by electrode and recording from other areas to see, when you stimulate an area, which other areas neurons are excited. That means they are connected to this area. Also, we can stimulate certain areas and see the behavioral response. Okay, we know that when the female bird listens the song of a male bird, certain areas neurons start to fire, get activated. So that is another way to understand that which areas are being activated in female birds by the male song. So we can temporarily or permanently inactivate the brain structures, right? And this is just one study where the lesion was made in NCM area, cordomedial nadopallium, and this structure is in the auditory pathway, that means listening, and inactivating or lesioning this area impair song recognition in zebra finches. Okay, they just used the speakers, and in the experimental chamber, they set up the speakers and broadcast the song of the bird's tutor. Okay, and then they compared the behavior of the bird with the broadcast that was presenting a novel zebrafinch song, so tutor song, as well as new song from same species. Before the lesion, the bird in both groups showed a strong significant preference for the song of the tutor. That makes sense, right? And after the surgery, the bird, one group that received lesion that did not show any preference. However, the sham lesioned operated group maintained their preference. So that tells that this area, NCM, is responsible for the recognition of the tutor's or parent's song. However, was intact, was not interrupted or altered by the lesion. So this NCM is not related to the production of song, but it is related to the recognition of the song. And here in these histograms, you can see the preference score for tutor song, and they measured the preference by the behavior of the listener zebrafinch, and here you see the control and lesion. This is pre and this is post lesion. Okay, so you can see the difference. So this is just one example how the experiments are done. Okay, now as I mentioned that there are multiple structures, discrete structures are present in the brain of a songbird, and they are connected differently. There are 4 different pathways so far we know, and first I'll talk about 2 of those, then I'll talk about the other 2. So here in this picture you can see by 2 different colors, red and gray. You can see 2 different pathways. The red pathway is mainly in the frontal part of the brain, right? So this is called anterior forebrain pathway. Okay, anterior, that means the front, forebrain pathway, and the gray colored pathway is showing the motor pathway. Okay, that's for the production of the song. Okay, so this is anterior forebrain pathway, red, and this is motor pathway. Anterior forebrain pathway is necessary for song learning and plasticity. So this is responsible for the learning and plasticity. Okay, and motor is responsible for the production of the song. That's why you see the motor output is finally going to the respiratory muscles and syrinx. As I mentioned, syrinx are lung-like structures in the bird, and specialized structure for the song production. So if you see the motor pathway, you see here that higher vocal center, HVC, projects into RA here, and RA is the abbreviation of the structure that is called the robust nucleus of the arcopilium. Robust nucleus of the arcopilium. Okay, and then RA sends signal or axons to the separate motor nuclei, where the motor neurons innervate the vocal organs in birds, okay, called the syrinx and the respiratory muscles. So you see the output from RA is going to these two separate nuclei, and then from there, from one nuclei, signal is going to the syrinx, and from another going to the respiratory muscles to produce the sound. Okay, vocal output. So that is the motor pathway, right? Now if you see the AFP and anterior forebrain pathway, you see that an area that is called area X. Okay, it's an important area, and this area is homolog to the mammal basal ganglia, mammalian basal ganglia, area X. Okay, in human we have the basal ganglia, which is similar to the area X, and then another area is DLM, this area, and the area DLM is the medial portion of dorsolateral nucleus of the thalamus. Okay, so dorsolateral nucleus of the thalamus, which portion? The medial. So DL, dorsolateral medial of the thalamus. Okay, and another structure in this pathway is L-men, the lateral subdivision of the magnocellular nucleus of the anterior nidopallium. So anterior nidopallium, this one. Okay, I'm repeating again, L-men, that indicates the lateral subdivision of the magnocellular nucleus of the anterior nidopallium. Okay, and from L-men, the signal is actually you see that the signal from L-man goes to the RA. So this pathway is connected to the motor pathway, which makes sense, right? So after learning signal, that signal should go from the memory system or template to the motor output. So these are two distinct pathways. One is for the learning and plasticity formation, another is for the motor or vocal output, right? Now, as I mentioned before, that studies deactivating parts of the neural circuit for songbirds via lesion tells us the functions of different areas, right? While you make a lesion of L-man or DLM or area X, what we see lesion in these areas in adult, it shows very little effect on already learned songs. Okay. However, if you make lesion in any of these areas before song crystallization, that leads to abnormal song. So the birds, before the songs are crystallized, if you inactivate these areas, then the birds, the pupil or learner will produce abnormal songs when their learning period is over. That tells that that learning and plasticity, which is the stage before crystallization, are controlled by these areas or this pathway. Okay. Now another experiment showed that lesion of area X in juveniles prevent crystallization, whereas lesion of L-man in juvenile results in permanently crystallized atypical songs. Okay. So you can further study the individual functions of these areas, and they have specific functions, right? So if you inactivate area X in juveniles, that prevent the crystallization. Okay, crystallization will not happen. Okay, whereas if you make a lesion of L-man in juveniles, that will result in permanently crystallized atypical songs. That means the song will be atypical, not like parent song, and it is permanent. You won't be able to change it. So permanently crystallized atypical song. Okay. So these are two important pathways. However, there are a couple more pathways in the songbird's brain. Okay. So here what I mentioned, just to go over, and you'll get an idea how these areas are responsible for processing the song, and if you inactivate or dysfunction these areas, then how that impact the song production. Okay. this is something interesting. I mentioned that when make songbirds come close to the female, the HVC, higher vocal center area, becomes activated and the bird starts to sing. Okay, so and lesion of this pathway in adults or in juvenile makes affect the song production. Okay, males affect the song production, of course, because this area is the motor output area, which is responsible for the production of the song, not the learning. Okay, there are two other pathways. One is the auditory pathway, and this picture is more complicated than the one I showed you before, because here you can see by blue and red those two connectivities I showed you before. However, you can see here this, gray and green. These are two other connectivities in the brain of the songbird, and there is another small one here, the yellow one. However, these two are also really very important. Okay, one is the auditory pathway, that is CN, LL, MLD, thalamic OV, L area, and NMC. So you can see this is NMC, CM, right? You can see OV, MLD, and CN. So this is CN. So these structures are in this auditory pathway. Okay, and this pathway is sensitive to auditory input. Okay, so when the pupil listens the parent song, these structures are activated to acquire that song. So this area is responsible for processing the auditory inputs as well as memorizing and recognizing the tutor's song. Okay, and deletion of this pathway causes disability in the adult bird to recognize the tutor's song. That experiment at the beginning I showed you the histograms. Remember the behavioral studies, sham lesion and lesion groups, and how that affected the behavior or response to the tutor's song or novel songs, non-tutor songs. So if you make lesion in this pathway, then the memorization, right, learning will be affected, recognition will be affected. However, once the birds already cross the crystallized period, then if you cause lesion, that will not impact. Okay, this pathway is important during the critical period when the birds listen and memorize the parent song. Okay, so listening and memorizing are the main functions of this area. Okay, subthalamic pathway is another pathway that includes nucleus UVA and then forebrain nuclei NIF and higher vocal center. So higher vocal center is actually a part of multiple connectivities. So you can see that signal from many different structures go to higher vocal center. HVC. And anyway, this subthalamic pathway is shown by this green pathway here. Okay, and this pathway is important for defining the sequence units sung within the song. So you know that within the song you have the notes, right? The smallest units, as I mentioned. How the notes will be sequenced within the syllables or motifs. Those are actually planned by this pathway. Okay, sequencing. If you just randomly sequence, that will not produce a beautiful song or sound. It would be like if you ask somebody who never learned piano, right, or violin, if you ask that person to play, that person will play randomly. That won't make any beautiful melody. Same way, and if you ask someone who already know how to play, then when that person will play, he knows how to put the notes and in order, and that will produce beautiful melodious sound. Okay, so same way, that's why this area is very important. Okay, now after the pupil or learner or child completes learning, they get separated from the parents. So it is very much like the human life. After the kids they'll get independent, they get away from the parents and start to build their own territories, own circles, right? And the birds do the same way. The birds, songbirds, after they finish learning the songs, they would build their own territory. And interestingly, this is very interesting that once they get separated from the parents, they use the parents' song because that got crystallized. However, studies have also found that they start to add some components to make the song more melodious, more beautiful. So it is like adding some artwork, additional artworks to make the painting more beautiful. So this is something very interesting finding in birds, songbirds. They try to be more creative and distinctive from the parents. Another very interesting finding about the songbirds learning is a group of scientists from the University of Chicago, who actually the PI and the main person who worked on this project, and he found out that during the sleep, the songbirds brain practices the song, so refresh their memory during the sleep. How they know that? They implanted the electrodes, right, in the brain, and they found that when the birds are in sleep, completely in sleeping status or stage, the certain brain areas, those areas we discussed, some of those areas, those are responsible for the memory, get activated when they are sleeping. And that is something interesting that uh, in old times parents used to say. When I was young, my parents used to say that sleep well before the night of your exam or test. That will make your memory better. So that makes sense, right? So this person who discovered this worked with in Professor Suga's lab, where I also worked. He was a postdoc there too, and then he started his own lab at the University of Chicago. So understanding the neural pathways and connectivities and the functional areas would help us to understand how the human baby learns the song and learns the speech and produce the speech, and that would also help us to understand the underlying mechanisms of speech disorders. That many children suffer from delayed learning of speech, or slow learning, or even abnormal speech. So we will understand the mechanisms better if we understand the songbird's brain. Okay. Now I'll talk about the gene responsible for the speech production. If you see the human as well as the songbird, a very specific sets of genes are present in both the human brain and the songbird's brain. Okay, and the most prominent gene is FOXP2, which is found in many vertebrates, um, where it plays an important role in the birds, songbirds, and also in echolocating bats. This gene has been isolated or identified. FOXP2 is required for the development of speech and language in humans. Okay, so as I mentioned, this is a very prominent gene that has been identified in the songbird, certain structures, and as well as in echolocating bat's brain, as well as in the human brain. In those areas are responsible for the speech and language. Okay, and studies have also shown that in human, mutations in FOXP2 cause the severe speech and language disorders. Okay, and also studies of the genes in mice and songbirds that indicate that this gene is necessary for vocal imitation. Okay, so this is a very important as well as interesting finding. Okay. This gene was first discovered to be associated with speech and language in the sequencing of a British family who the members had been suffering from the speech disorder, and from there sequencing this mutation of this FOXP2 was first discovered. Now many groups are working together combinedly to create the mappings of the genes. Those are responsible for speech, and the mutation of which one is responsible for which type of speech disorder. Okay. Now, besides songbirds, another kinds of birds, those are also very interesting. They learn how to mimic a particular sound very effectively. One thing is interesting, that songbirds' brain learns how to produce the parent's song. It doesn't like the other songs. That means it won't like the songs from other species. It won't prefer to learn the songs from other species. It will learn the song from the parents and same species. However, certain birds, their brain is designed in a way that they can mimic or imitate any sound, including the human voice. For example, parrots. Right. I'll post a very interesting video that certain species of birds, how beautifully they can mimic the sound they listen. They can produce the sound of a camera. When you shoot pictures, the sound of it very precisely, and or human voice, or any other type of sound, they can quickly learn and reproduce. Okay, so it is very interesting. And also studies are being performed to understand the genes. Those are responsible for the mimicking or imitating the song, imitating the sound. Okay, so just go over this couple of slides, and the important things I have already explained. So in this part, you need to understand the difference between the innate template and acquired template. Innate is like by nature, how a bird flies. You don't need to teach the bird to fly; they will get or that ability by birth, because their genes will detect that and give that ability. Okay, how a fish swims in water, how a human walks, right? Moves their its hands and legs and body. All these are innate. By the movement of a tiger, you can even if you don't see which animal is that, if you just only see the movement, that way it is walking or running, you can tell that this is a tiger. So that is the innate template. Because of the innate template. Acquired is like the behavior we learn by training and learning from the nature. Also, you need to know those 5 stages of song learning, creating the solid crystallized songs, and also you need to know those 4 connectivities or networks, and the important areas only those are present in those pathways. Okay, so everybody, let's stop here.